Hello, everyone. Thank you for joining today's S-CHOice webinar. My name is Janet Lee. I'm the Senior Director of Cell Line Development Team at Samsung Biologics Antibody Development Center. It is my great pleasure to be here to share our insights for the latest innovation in cell line development technology for rapid biopharmaceutical production. Today, I'll discuss two cell line development platforms, S-CHOice and S-AfuCHO, that Samsung team has developed to provide timely solution for complex biologics development program. So with rapid expansions in biologics market, many complex molecules are filling in our development pipeline in recent years, adding additional complexity to CMC development program. In this webinar, I'll highlight how Samsung Biologics cell line development platform called S-CHOice and S-AfuCHO can address these development challenges with improved efficiency. Throughout this session, we will also discuss industry trend, CMC development insights to accelerate development process to best leverage our cell line development technology platform. So before we dive into the details of this platform, let's take a moment to understand the evolving landscape of biologics development platform and the key trends that are driving these innovations. Just name a few: rising complexity of biologics. Biopharmaceutical pipelines are now featured more complexity, such as a complex molecule, bispecific, multispecific antibodies, or antibody-drug conjugates, or cross-map, not to mention the fusion protein. This format presents unique cell line development challenges, including difficult to express or clonal instability, or difficult to control post-translational modification for efficient product quality control. The second note that we would like to address is the accelerated timeline. No matter how complex it gets, with increasing competition, the need to shorten the development timeline has become more critical than ever. Accelerated clinical material generation and seamless scalability to commercial manufacturing are essential to keep the intended IND target timeline. Therefore, scalable and streamlined cell line development platforms are critical importance during CMC development needs. Thirdly, the stringent regulatory expectations are also raising bar for safety and efficacy of patients and consistent biologics program. Therefore, robust cell line development process are the key requirements to ensure quality compliance with international regulatory standards. In addition, quality beyond quality productivity. Therefore, optimizing therapeutic functions like antibody-dependent cellular cytotoxicity, also known as ADCC, or glycosylation controls has become as important as achieving high-yield process for the COG concern. Lastly, how about the sustainability? Sustainable practice, including energy-efficient process and waste reductions, are becoming key factors in biomanufacturing environment. So to meet tighter product production in CMC development needs, we have introduced lipid transposase workflow in our S-CHOice cell line development platform for the past 2 years, and we would like to cover the transposase technology topic next. So as shown in the figure 1 image, the transposase system contains the flanked region of gene of interest with inverted terminal sequence called ITR, so ensuring precise targeting and stable gene integration into the target genomic loci. Therefore, transposase will carry insert transposon vector into target recipient site of active chromatin sequence by cut and paste mechanism, resulting efficient delivery. As a result, much higher productivity was achieved in our S-CHOice workflow utilizing a transposase system due to increased gene integration efficiency, leading to high performance clone generation. So what about the transgene integration efficiency using transposase system? As shown in my data, much higher gene integration efficiency was achieved utilizing transposase system in our S-CHOice cell line development platform. The figure 3 compares 3 case studies. As shown, case ABC highlights a random integration versus the transposase platform. The final clone titer performance at 14-day fed batch study showcases much higher titer productivity throughout the transposase system when compared to traditional random methods. These data demonstrate improved probability of finding high-performance clones using transposase workflow against random traditional methods. Greater than 75% of high-performance clones were generated utilizing transposase workflow, showcasing improved efficiency and also improved timeline. Next slide. So the improved productivity was achieved across the evaluation group, case ABC. The figure 4, as you can see, titer improvement, but also figure 5 highlights the product purity improvement across the case ABC. I am sorry to mention case A is the difficulty expressed modified map. Case B and C are complex molecule with more than 2 vector systems. As shown, the percent purity by CESDS demonstrates much higher improvement after first step purification when transposase system is employed. So therefore, these data demonstrate improved probability and improved quality across the transposase technology implementation. So just name a few. The transposase approach provides several advantages as no limitation to gene of interest delivery size, faster recovery upon transfection, and faster scale-up process because this gets plugged into our platform process all the way to cGMP manufacturing up to 15K, and no time is wasted for early material generation to meet your competitive CMC development timeline. So in summary, much higher productivity was achieved using our S-CHOice transposase due to increased gene integration efficiency, leading to high-performance clone generation. Also, cell line development timeline is something notable to mention. Our monoclonal antibody development timeline from transfection to MCB top clone selection is now reduced to 2 months. And if you should have complex molecule with a multi-vector system, it would only take about 3 months from transfection to MCB generation candidate selection. So I think this is a notable advancement in our cell line development platform. Now I'm excited to introduce additional platform that we have newly launched this year. It's called AfuCHO S-CHOice host cell line. So why S-AfuCHO cell line was developed? As you know, the presence of core fucose moiety on antibody Fc region can inhibit antibody-dependent cellular cytotoxicity and therefore reducing antibody therapeutic efficacy. As a solution, Samsung presented AfuCHO-free S-CHOice cell line is a new cell line that we have introduced to build up upon our track record. Since AfuCHO has been demonstrating to increase ADCC activity in both preclinical and clinical settings. So we have demonstrated clear track records. We have conveniently just removed the footage genome from existing S-CHOice host cell line to deliver efficient ADCC program. So for AfuCHO cell line generation, we have engineered the footage genome from current S-CHOice host cell line, which was introduced since 2020. So we have all this track record utilizing the cell line. And in terms of the cell line productivity, performance evaluation data demonstrate in this figure 6, the comparable cell growth and productivity was achieved when S-AfuCHO cell line was transfected with a reference monoclonal antibody, demonstrating comparable stability, productivity across the control and S-AfuCHO cell line. The figure C demonstrates our stability. We have demonstrated across the top 3 clones, across the 60 population doubling. Left side highlights our S-AfuCHO newly developed cell line versus S-AfuCHO S-CHOice control host with a genome in it. As shown, it's very comparable across the study group, demonstrating it is very stable across the study groups. So next, figure 7, next slide contains the absence of the core fucose. As shown, fucosylation level is near 0% for S-AfuCHO, whereas the control S-CHOice still maintains a relative amount of fucosylation. Figure 8 demonstrates the ADCC activity. In terms of ADCC activity, significantly higher ADCC activity was achieved using our S-AfuCHO cell line. Transfected with a reference map, this case was IgG1 type trastuzumab compared to S-CHOice control cell line. I want you to pay attention to green data set. That's our S-AfuCHO. As you can see, significant improvement in ADCC activity, showing higher potency for ADCC program. So you can benefit, enhance the ADCC activity using our S-CHOice fucose-free cell line with high productivity with confidence. So in summary, Samsung Biologics platforms are strategically prepared to meet your complex biologics development needs. Our S-CHOice cell line development platform offers optimized cell line productivity, scalability, and speed to IND through either transposase technology. Our S-AfuCHO cell line enhances ADCC therapeutic efficacy with a fucosylation-free cell line, offering superior ADCC activity while maintaining high productivity. So with that, at Samsung Biologics, we are investing in scientific innovation to provide efficient development solutions to create predictive development platforms, regardless of your modality, to provide successful development process. So please leverage our efficient development platform and manufacturing expertise. So thank you for your attention, and I look forward to have your further discussion for questions. Thank you.
So our first question is, can S-AfuCHO be adapted for non-antibody-based biologics?
So while S-AfuCHO cell line is engineered in Fc region to increase the ADCC activity in antibody, its fundamental technology can be explored for other biologics as well. So currently, we're actively researching applications for broader therapeutic modality. Our fucosylated antibodies are intensely used in the current market to advance medicine due to their high ADCC program. This makes an effective binding to specific targets such as immunotherapy and autoimmune disease as well. I hope I answered your question.
And it looks like our next question is, how do these platforms handle regulatory challenges?
Sure. Both platforms are designed with regulatory compliance in mind. So for instance, S-CHOice ensures traceability and consistency during entire cell line development process. And it's been launched since 2020. So we have close to 5 years of a track record. And to date, we have up to 36 IND approval utilizing our platform. Therefore, S-AfuCHO aligns with the current guideline to enhance therapeutic functionality without compromising safety and quality of the cell line.
Can you share insights on timelines for implementing these platforms?
Sure. The timeline varies based on complexity of the client's molecule. Our S-CHOice platform currently offers as early as 2 months for monoclonal antibody. This time includes transfection to MCB candidate clone selection for MCB banking, and about 3 months for complex molecule program with more than 2 vector systems, which can significantly shorten development timeline, which was much longer prior to transposase implementation.
The last question for the webcast is, How do your platforms compare with CRISPR-based cell line development technologies?
Oh, yeah, wonderful. So while CRISPR-based technology presents precise genome editing tools, there are still many hurdles to overcome, such as the high cost burden during the drug development process. And many clients are very conscious of their development costs. So our S-CHOice cell line development platform offers simple solutions by offering cost-competitive strategy, utilizing our talent technology to better support client development pipeline. So more drug programs can see the development opportunity to reach their patient faster. So with our Transposase technology and our S-CHOice platform offers high production titer needs to really improve the C of G concern for our clients. Thank you.